Good afternoon to all the participants present here. I welcome you all on behalf of the Department of Biotechnology to the National Webinar on Marine Research and Aquaculture. It is a lecture series that will be there till 28th July. So we have eminent speakers from all over India talking on the different aspects of marine research and aquaculture. At this outset, I would like to sincerely thank our management, Dr. Maria Zina Johnson, our Chancellor Ma'am, and Dr. Marie Johnson, our President Sir, for providing us this platform to conduct this webinar series. It is also my duty to thank the advisors, Dr. Sasiprabha, Dr. Logashanmugam, Dr. S.S. Rao, Dr. Sundari, and Dr. Igni Sebasti Prabhu. A special thanks to Dr. Wilson Aruni, our Pro Vice Chancellor, who has been instrumental in bringing out this webinar. Today is the first day of this webinar series, and we have a speaker, Dr. M. Arumugam from CAS in Marine Biology, Annamalai University. Sir will be giving out a talk on drugs from the sea, a voyage. So, Dr. Arumugam is specialized in the field of marine biotechnology and marine natural products. He has an experience in teaching and research for around 16 years. He has guided 13 Ph.D. students, of which 9 of students have been awarded Ph.D. He has also guided MPhil students. He has various awards to his name, including the DAD Summer Trainee Award, the DST Travel Grant Awardee, NUS Visiting Fellow. He has awards from Sweden. He has a Best Researcher Award in 2016 and 17. Also, he has various projects to his name funded by MOES, UGC, DBT, DST. With a number of publications to his credit, he has a very good citation and H-index, which shows his research capability. Sir has visited countries like Singapore, Germany, Sweden, France, and has ample amount of experience in the area of marine natural products. I request Sir to present his talk today. Sir, please welcome, sir. Sir,
thank
over
you,
to you,
Dr. Ramesh Kumar. Yes, thank you, Dr. Ramesh Kumar.
Yes, sir.
And for very good welcoming me to this wonderful webinar, which are organized by the Sathyabama Institute of Science and Technology. And also my special and sincere thanks to my senior, Dr. M. Maslamani Selvam, who is the organizing secretary of this program. And also, I should thank the authorities of Sathyabama Institute of Science and Technology, Chennai. And today, I'm going to present, the title is Drag from the Sea, a Voyage. My dear friends, are you here?
Yes, sir, we can hear you, sir.
Fine. My dear friends, as you know, the ocean is provide lot to us in terms of food, in terms of energy, in terms of medicines, in terms of current, etc. Similarly, this is, all are very well known about this pandemic period, and this is very challenging situations in all over the world. And the searching of drugs, not only for the corona virus, the searching of drugs in national as well as the international level is very long history. And especially in India, Ministry of Earth Sciences, Government of India has created a separate platform for drug from the sea program. Almost all over India, 14 research institutions which are very much involved to find out how we can developing a potential drugs from the sea. Because the India is very vast coastline, around more than 8,100 square kilometers, which are covering by the coastlines. So that's why the source of the drugs is very, very vast. And also the ocean almost covering all the east coast, starting from the west to east coast, our country, which are covering almost all the coastlines. and maximum starting from the Coromandel coast to east coast of India. So plenty sources which are available for providing medicines, chemicals, or some other materials. What are the materials which are available in the marine? It's very, very unique when compared to the other terrestrial organisms or the terrestrial counterparts. And my dear friends, now I'll go to my presentation. Please, 1 minute. I just share my screen. Then I will discuss one by one. So the title of my today talk is Drug from a Voyage. My dear friends, this is my university. Almost all of you very well know about the university. It's a state university located in Tamil Nadu, established in 1929. And also we have the lot of ranks and recognition by the Times Higher by Harvard University, as well as we have the rank at 11th amongst the India top 10 collaborating institutions by the Nature Index. So Annamalai University with 49 departments of study in a single roof. And also the Annamalai University has among the research institutions in the field of chemistry, pharmacology, research and standing 17th rank of for publications in all the subject areas from 2002 to 2013. And we have reached the Scopus Index by the National Science and Technology Management Information System, Department of Science and Technology, and the Ministry of Science and Technology, Government of India, New Delhi. And the center is called as the Center of Advanced Study in Marine Biology, shortly called as Biology. It's for marine sciences in India, and it is only one center as an advanced study for marine biology since 1963. What are the recognition for our center? The is Ministry of Science and Center as Center of Excellence in Ornamental Fish Culture 2004. And Department of Biotech identified our center as, so this is the aquatic center. Almost my seniors and other classmates there were known because they are studying. This slides is not necessary for our classmates, but it's somewhat new to others. And I already told you that's a lot of the recognitions for our center. And also this is the center has been striving hard to rise standards to an international platform. And in India, some of the universities or the institutions have been focusing on the marine sciences and related research. This is dedicated towards research on marine natural products and drug discovery is to be addressed. As such, a center not only delivers ample opportunities to the students, fraternity, research domain, but also the researchers for the development of drugs for various diseases prevalent into the society. Finally, the center creates an environment that ensure a balance between the marine environment and explain or explore the bacterial compounds for the drug developments. My dear friends, the drug from the sea, as you see this slide, this is a fish, particular fish called as the lionfish. That means the marine is that we are gold mine on our earth. What you want? You want food, you want medicine, you want energy, anything else. The ocean is ready to provide us. The only thing is we should explore the organisms, explore the sources for the right point of view. So that is very much important. And when we are discussing the drug from the sea, we should know the marine natural products. The marine natural products, nothing but what are the chemical compounds which are produced by the marine organisms called as the marine natural products. The bacterial molecules from marine sources find extensive traditional use and predominantly as a crude form and also helping as templates for synthetic preparations. And the world market value of marine derived drug reaches around 4.8 billions in 2011, and it should be raised from 5.3 billions US dollars in 2012, and it will be reached 10.6 billion by 2019. And marine environment is a harboring and wealth of natural products through the producing the pharmaceuticals, nutritional supplements, enzymes, and other cosmetics. Why the marine organisms the natural product of the molecules? Because the marine organisms or the marine environment, it is very unique characteristics of marine life. It's due to the rich biodiversity, adaptation, predations, change environment, communication, and signal. And all these categories is somewhat very unique. So that's why the marine organism having lot of adaptations. And also the most of the marine organisms are from the environment. That's why they should synthesize the unique metabolites or the molecules for survival on the environment. And also the biodiversity. Almost the biodiversity is very vast when compared to the land. And this is the marine natural products as well as the biomaterials which are available are produced from the marine sources. And my dear friends, the compounds which are classified into major groups, the carbohydrates, fats or the fatty acids, as well as the nucleotides. So in the case of ceramics, which are dealing with the development of biomaterial from the sea, but remaining all the four categories like the carbohydrate, protein, fatty acids or the fat, nucleotides, all these things which are responsible for producing lot of molecules for the various applications, especially the chondroitin sulfate as well as the chitin and chitosan. This is a very unique molecules which are plentifully available on the marine sources. Hello? Hello?
Yes, yes sir, you can continue.
Yeah. Okay. And what is the depth of these compounds? How they can derive? How they can produce? That means marine diversity, including the three important categories. First, starting from microbes, then flora, as well as the fauna. So in the case of microbes which comprises bacteria, fungi, and actinomyces. In the case of flora, macroalgae, seaweeds, seagrass, and mangroves. So these are the major this flora. And in the case of fauna, group which consists of invertebrates as well as the vertebrates. And these are the chemical classes of marine natural products. What are the major chemical classes of the marine natural products? Starting from the aliphatic, alkalides, carbohydrates, terpenoids, amino acids and peptides and others. So these are the major classifications. And what are this slide shows the natural products from the marine flora, starting from the algae to the seagrass. Because the algae or the seaweeds, they contribute a lot to us in terms of the food items as well as a lot of medicinal products. And similarly for the other phytoplankton groups. Natural products from the marine microbes, this is very much important, which are involving by the bacteria, fungi, and actinomyces. And this slide shows the marine natural products from the fauna. In the case of faunal diversity, starting from the porifera, cnidaria, echinoderms, chordate, and mollusca, they have to contribute a lot of chemical substances, which including the terpenoids, alkalides, aliphatic groups or the aliphatic molecules, steroids, carbohydrates, amino acids, and so on. And this is the information of the slide shows the diversity of invertebrate phyta. When compared to land, the marine invertebrate diversity very vast. There are phyla which are available, of which 10 phyla exist in terrestrial environment and 22 phyla living in the sea, of which 14 are exclusively on marine. So the major marine phyla are shown in the, especially in the arthropods groups. That means the crustacean forms. And these are the compounds, list of compounds which are synthesized by the marine microbes, various activities, starting from the blood pressure and as well as the supplement material, and also a lot of cosmetic products and food additives, antioxidant material, agricultural materials, just like organic manure, and even we have produced some of the energy from the microbes. And this slide shows marine algae. As you know, the seaweeds classified into 3 major groups, starting from the green, brown, and red algae. Maximum all the seaweeds, they have to provide lot of compounds or the molecules. Most of the compounds, such as the laminarin, other fucoidin, polyphenols, or even the methanolic other extracts and flavonoids, all these things which are responsible for the cancer. So most of the marine seaweed source are the seaweed molecules which are very much responsible for anti-cancer activities. And also, this is the one slide shows the potential of the seaweeds. The particular seaweeds, we can provide number of compounds or number of materials, starting from lipids, fatty acids, cherals, protein, phycobil proteins, phenolic components, lot of other antioxidant molecules, carbohydrate, agar or agarose. In addition to that, the marine natural products, the bioactive compounds, is from the marine organisms also. Lot other waste material. What are the waste materials which are comes out from the sea organisms while catching the fish? Example, their skin, their what is that? Their skin. Everything it's dumped into the sea, it's considered as a waste. But as a marine biotechnologist, we have to develop a product, very much useful product from the waste at the concept of. My dear friends, sir, you told that the lot of sources which are available and we can derive all the organisms very easily. No, it is not possible, because when you are aware and beware, dive on deep. That is very much important, because lot of venomous fishes or the venomous organisms which are available on the sea. And starting from the slide shows the lionfish as well as the ray fish. So both of the fish, they have provide very good analgesic compound or the analgesic molecule to the nature. And the slide shows the orphoterine. The orphoterine is an analgesic agent, as well as they can act as a pain relieving agent from the ray fish as well as the lionfish. And for example, the venom components, the venom is nothing. What are the materials? That means the compounds which are synthesized by the animal. Just one drop of venom, which consists of more than 25,000 of molecules. So what are the major molecules which are present are the venom components? Starting from the bradykinin, this is a list of compounds or the molecules which are available. Starting from the bradykinin, hemolysin, serotonin, histamine, prostaglandin, hyaluronidase, nucleosidases, alkaline protease, acid phosphatase, fibrinolysin, number of the enzymes, number of compounds or the molecules which are synthesized by the drop of venom, which are secreted by the marine animal. And my dear friends, you just see the slides. What is the potential of marine organisms in terms of lethality or mortality?
Cone snails are marine snails found in reef
And
environments throughout the world.
conus knights, which are coming under the marine gastropod
All
mollusks.
species of cone snails have
They
a highly
are very
sophisticated
small
venom
mollusks,
production
and
apparatus and delivery
they are
system.
very sluggish animals.
The venom is produced in a
At the
tubular
same
duct
time,
that is often several
the
times the length of the
mollusks,
snail
which
and
consist
is
of
attached to a muscular bulb.
venom
Hollow, spear-like
sac,
teeth are
as
made in a
well as
radular
venom
sac
bulb,
and filled with
having
venom.
the venom ducts. It's a very long venom ducts, and also
In
having
fish-eating
the
species, the cone detects its prey using
injecting organs.
chemosensors in its
And you
siphon.
can see how this sluggish animal, they can easily catch the very
Upon contact with
fast
the
swimming
prey,
animal
the
or
proboscis
swimming animal
impales a
like a fish.
harpoon-like tube in any exposed
Just
tissue,
a drop of
injects the
venoms
venom,
which are
and paralyzes
very much
its prey.
necessary to kill the organisms. Once the animal is killed, then they can easily swallow. This is a way the toxins which are synthesized by this particular animal, which are very much helpful for catching the prey for their food purposes. And you can see after injecting the toxins to the fish, they can immediately paralyze. That shows the potential of compounds which are synthesized by the conus. And my dear friends, then you can ask a question, sir. So any other recent developments which are available on the marine nature products research, sir? Yes. Lot of information which are available, and these are the major groups which are involving in the drug discovery regime among the 34 marine phyla. And starting from the chordata, arthropods just like the crabs, prawn, etc., and nidaria such as the jellyfish and mollusks, echinoderms, and poriferous. And this is the list of anti-cancer compounds which are derived from the marine organisms. Even these are the organisms which are providing a very good anti-cancer compound. And most of the compounds, they have successfully completed clinical trials. Now it is available in the form of Yondelis, Halavan, and these are the slides shows the antiviral and the analgesic agents of the molecules. There is the withdrawing, zincnotide, and omega-3 acid ethyl esters from the lovosa, available in the form of lovosa. And especially the zincnotide available in the form of prialt, which are very much necessary or which are very much useful or alternative for the morphine. My dear friends, those are the patient is very much affected by the cancer or AIDS, doctors immediately prescribe the drug as the morphine to the reduce the pain. But the morphine shows the lot of side effects. But when compared to the morphine, these alternative drugs which are derived from the marine organisms, they shows very potent and also they doesn't cause synergistic toxicities. And my dear friends, these are the molecules. That means 1-orthohexadicyl glycero 3-phosphocholine ester, which are derived from a mollusk. That means the nudibranch mollusk from our center at my laboratory. This is very potent agents for the anti-cancer drug. And this is the slide shows the. You can see how the marine organisms available or act as just like a predators. And you can see the toxic or venomous spines which are present on their tail regions. And in this slide, this part shows the, you can see their spine. And you also see the arrangements of barbels. And these are called as the barbels. And you can see how the barbels which are uniquely arranged in the opposite manner. Once these spines which are entering into the victims, what happens? This entering into the victim, and once you have to remove the these available barbels are retained on their body. That means the victim bodies. So it leads for a lot of inflammations. So that's what the immediate responses, very severe pain, and sometimes the mortality is happening. But they show the painful lessons, but also they have to provide very good drugs, very good molecules, especially the analgesic as well as the anti-cancer molecules from the venom components. And my dear friends, this slide shows we have recently studied the potent coagulation venom from the sea snake. My dear friends, the sea snake is a very dangerous animals which are available on most toxic animals which are available on the earth. Because in addition to the tetrodotoxins from the puffer fishes, this type of the fishes, they have to produce very unique molecules or venom components, and which is responsible for the coagulations. So once the blood is coagulates, definitely you should know what is the impacts of the coagulations of the blood. Immediately, the entire circulatory system which are affected. So it leads for immediate mortality. So we have identified what is the protein and what is the molecular weight, what are the name of the peptides, all this kind of information we have derived. And you can see in how we can collect the venom from this sea snake. And this work is did at my laboratory. And you can just take a snake, and you can if you are giving the gentle press on their head regions, they can milking their venom immediately. So at this tip, you can just placing a end of tube, and you can collect the venom. And this is the molecular weights of these isolated venoms which are derived from the sea snake. My dear friends, and after the study of sea snake venom, we are not only studying the sea snake venom, we are also concentrating on some other molluscan group of animals. This is the animals, sepia, which are producing the collagens, especially the type 1 collagen. And as you know, the collagen is very much available among all the animals. But in the case of type 1 collagens, which are only present in the placenta. So that's what the type 1 collagens attracting much attractions by the researchers. And also they have a lot of the medicinal aspects of the medicinal properties. And after the isolating of the type 1 collagen, we have derived, or we have making different biomaterials such as the film as well as the gel. And after production of the biomaterial such as the gel as well as the film, then we have introduced some of the antibiotics and what are their drug delivering efficacies by these biomaterials which are synthesized by the sepia. So this kind of studies is also studied. This information is available on this a very good journal, Process Biochemistry. And my dear friends, the next slide shows, and this is the some of the biomaterials which are derived from the sepia, the cephalopod group of animals. So after loading the antibiotics in the gels as well as the film, the slide shows how the animals once they affected in wounded animals, how they can recover when we are giving the dose to the animals. So almost within the 10 days, almost all their wound is closed from the initial stage to the left side slide and to the right side slide. My dear friends, so these are the previous stories about our center as well as my laboratory. And I already told you that the Ministry of Earth Sciences identified 14 institutions in India, and Center of Advanced Study in Marine Biology is one among the center. And Ministry of Earth Sciences extended the support for 2 times with the support of our dynamic director, former dynamic director, and my mentor, Professor T. Balasubramanian sir. And now this program is going on, and we have developed 24 different lead molecules for the anti-fertility, starting from the anti-fertility, anti-malarial, antibacterial, and anti-HIV. So these are the reports which are still available, and we have submitted the report to the Ministry of Earth Sciences for developing the lead molecules from the sea. And also this is a very major achievement from our center, especially for the drug development research. Apart from this, we have produced the low molecular weight hyaluronic acid from the mollusks. My dear friends, but anybody knows about the hyaluronic acid? Actually, the hyaluronic acid is just like a synovial fluids, which are available on all our joints. Once the concentration of the hyaluronic acids, which are reduced, what happens? It leads for arthritis. It leads for arthritis. So that's why, especially the low molecular weight of hyaluronic acid play a major role. So that's why we have targeted the low molecular weight of hyaluronic acid, which are derived from the marine mollusks and their properties, all these things. And in addition to that, some of the fucoxanthin extract or the purifications were did at my laboratories, and these are the chromatographic analyses were performed at our center, and maximum all the fractions shows very good potential, very good activities in terms of anti-hyaluronidase as well as the anti-tyrosinase. Why we are studying the anti-tyrosinase as well as the anti-hyaluronidase, we will discuss. So, my dear friends, what is the applications on these things, especially the fucoxanthin? So, these are the normal skins which are shows here, the top. And what is that worries for once you are reaching the 40 or 45 plus? So, what happened? The aging process. We can't stop our aging, but when we are taking some of the seaweeds or when we are applying some of the fucoxanthin or other materials from the seaweeds, definitely we can reduce or prolonging the aging process. For example, wrinkles caused, that means the surgum. Wrinkles caused, it is due to the effect of hyaluronidase enzymes on the dermal cells. So that's why once if you are involving that particular process with the influence of hyaluronidase, definitely we will prolonging the formation of wrinkles. Similarly, that the tanning means that's just like a darkness. It is caused due to the effect of tyrosinase enzymes on the dermal cells. So once the tyrosinase action is happened, what it leads for the dermal or skin should be darkened. And so in that case, when we are taking the fucoxanthin, which consists of hyaluronidase as well as the tyrosinase, they may be act on both the cases, either the aging or the wrinkle formations as well as the darkness of the skin will be reduced. So we can in futures we can developing a cosmetics or the cosmetic material from the seaweeds. And my dear friend, this is the another important group of animals which are reared at my laboratory, rearing the green mussels. And this is in captive conditions. My dear friend, you should know, or most of the marine biologists or aquaculturists, they should know the shell or the mussels called as the Pernavirides. So this Pernavirides, what happened? They are the particular group of animals available as a cluster, just like a grapes. How it is possible? This particular animals, they can synthesize or they can producing a fine filaments or fine hairy-like projections. So that the developed fine-like or filaments-like materials which are helpful for attaching the rocky substrate. But our target or our aim is, what is the particular proteins which are responsible for making the particular filaments or hairy-like projections called as the threads. Our aim is how we can developing a biomaterial, especially for in the field of biomedical applications, or suturing, the open heart surgery, or even some of the delivering at the time of delivering, the opening the skin or opening this particular area. So in this case, the suturing is a somewhat painful or difficult process. In that case, if you are developing a biomaterials or the bioplaster, definitely just take the material and peel the product, and you should apply on this particular area. You will be clear. No need of sutures. So that's what we have planned, and almost we have success on this story. You can see how much the peptides which are available on this biceps thread, and this is the HPLC analysis of the thing, and this is a purification process, all these things, and this is already published in the International Journals of Peptide Research and Therapeutics. And also the processing the Stingray Venom, and recently we have developing some of the molecules from the Stingray. Stingray is very potent venom or toxic animals which are available on the air, sea, C. And I already told you that how they can synthesize the molecules, because they are having a very specialized the spines on the dorsal regions, that means the tail regions, and they have to secrete the secretor or synthesize the potent venom for getting the prey. And we have did some of the work on this aspect. We have collected the stingray from our course, and then we have processing the animals, and how we can collect the spines, and then we can go for the processing at the laboratory. And then the storage and homogenization process, every spines which are kept into their liquid nitrogens, and then we are processing further. And this slide shows the some of the, again, the collagens and their applications. And but this is somewhat different. This is not for the drug delivery applications. This is for the developing the collagens from the particular group of animals, and also we have developing the bone powder. That means raw bone powder from the other group of animals, and then we can developing the collagen as well as the hydroxyapatite. That means the hydroxyapatites which are derived from the bone of the marine animals like the ray fish. Then we can developing as the scaffold. So the scaffold, which consists of collagen as well as the hydroxyapatite for the bone tissue engineering, because as you know, the lot of accident and other natural as well as the man-made accident is happened, and most of the affected areas, our body, especially the legs and our hands. So that's why the bone replacement, it's also the need of our. So that's why we have to developing an artificial bone through the collagen as well as the hydroxyapatite, especially for the smaller area through their bone tissue engineering process. The work is going on. And my dear friend, this slide shows the this is what is it, sea urchin. And the sea urchins, you can see the number of spines which are available on the sea urchin. And once we have collected the animals, remove the spine from the sea urchins, then we can go for their what is the cross section of the particular spine of a sea urchin. And what a beauty is, when we are cross sectioning the spine of a sea urchin coming under the Echinoderm phyla, which are very much similar in the case of normal human bones. It's somewhat miracle. And when we are developing such kind of the sea urchin spines, that means the artificial bone in the sense, definitely we can repairing the particular area which are affected or damaged by the nature. So that's what we have planned, and the work is also the under process. And this is the recently we have prepared the hydrogels from the seaweeds, and after the collecting the seaweeds, and then we can developing the hydrogels. Hydrogels in the sense it is in the form of membrane, just like a carrier for the antimicrobial drug. We have evaluated the drug delivery purposes, especially for the anti-cancers as well as the antimicrobial activities. And this is also we have recently developed the drug delivering potential type 1 collagens from the eelfish also. That means we have developing the wealth from the waste, because while we are processing the eelfish, what happened? The entire skins which are removed and just throats. The remaining fleshy portions only consumed for food purposes. So, but what we are doing on this aspect, actually we have targeted or we are dealing with the skin only. That means waste material of this particular fish, and then we can processing, get the type 1 collagen for development of drug delivery applications. And this is another recent information. We have developing the bioluminescent bacteria from the marine sources, especially the some of the deep sea fishes at my laboratory. And bioluminescent bacteria or light producing bacteria that are predominantly present in the seawater or even the marine sediments, the surface of decomposing fish, and in the gut of marine microbes. And what happened? Our ultimate aim is we should develop not only the biomaterial, we should develop some of the glorious or luminous material, dressing material, which are very much attracted by the audience as well as the publics. So that's what we have developing a luminescent material or the luminescence items from the marine microbes, that means bioluminescent bacteria. And another recent findings and promises, my dear friend, then you can ask a question, sir, only the biomaterials are the drugs or the molecules which are synthesized by the organisms? You can give any other example in international level. In the sense, definitely, marine organisms not only producing or synthesizing the molecules, they are also producing the gold. This is very recent information. Might be some of them might be know about this story. And the gold, you see the photo gallery, which are produced by the bacteria. And according to the researcher at Michigan State University, and it's they can survive on the extreme toxic environment, and they can create not the 22 karat, they can create the 24 karat pure gold. It is possible. The bacteria is called as the Jellyfish acidivorous. What the process is behind on the screens? This part of self-defense mechanism, gold ions which are dissolved in the water, especially in the seawater, is somewhat toxic. So when the bacteria senses them, it releases a protein called delphtibactin A. So particular proteins which are released by the bacteria, then the protein acts as a shield for the bacteria to protect this kind of environment. And that proteins changes the poisonous ions which are available in and around the area of the bacteria into harmless particles that is called as, and that is accumulated the outside of the cells by the bacteria, that is called as the pure gold, just like a pop out. So this information is recently available in the international level, and this is another information. Doctors are trying and what is that unorthodox approach to treat burn victims using the skin. Brazilian doctors, they are taking an experimental approach to treat the burns using the tilapia skin. Just traditionally, after processing the tilapia, and just remove their skins and applying into the bones, which are treated using the pig or humans, and which are transfer the collagen, a healing portion, to their victim skins. And also the institute to turn to tilapia as an alternative in the community who suffered from the burns. And this kind of approaches is very much applicable for our soldier or even the army peoples also. That is possible. Why we are regularly using the conventional or side-effected medicines? We are thinking the alternative material for applying or removing, reducing their burns. And this is some of the tilapia skins be used as the bandage burns. Yes, nature are the natural materials which are reducing their burns. And my dear friends, then some of the other research institute in UK, they have developing optical features embedded on the marine shells to have developed a responsive or transparent displays. Because the unique animal, they're having a particular materials. What the researcher says that the natural optical structures may serve as a design. It is great for engineering color selective and also the controllable transparent displays that require in no internal light source. Just developing such kind of a biomaterial, you can apply even at your outdoor doors or even in the indoor doors, whatever it is, it's become illuminated. So this is another recent findings from UK. And another information is the green fluorescent protein GFP, which are derived from the particular jellyfish, I think jellyfish, and reveal the structure of pyramidal nerve cells in the cortex of a mouse brain. And particular GFP proteins which are act as a probe to reveal or to study the cortex of a mouse brain. Similarly, when some of the stroke is happen, some of the blockages happen on the arteries of brain, human brain also. Then if you want to analysis, diagnosis, definitely we could apply such kind of the GFP into the brain, and we have studied well. And my friends, and what is the general remarks, sir? You are sailing on the drug from the sea program, and what is the general remarks? almost the patents, what are the patents which are available on this particular area? Almost more than 500 patents which are available from the marine resources. Another maximum 50, more than 50% crude compounds which are derived from the marine sources. Almost 1,000 compounds available. That means available in the form of purified materials. And also what number of publications which are available on this particular area, more than 1,500 publications are available. And also these are the collection methods which we are regularly using for that. And this is some of the other international collection method, but or even we are attempting the scuba diving and as well as the snorkeling for collecting the marine organisms, and somewhat we don't have such kind of the automated research facilities for collecting the marine animals, but we are we get the support from the local fishermen, as well as we have some of the small seaweed research vessels. We can go and collect the animals for our purposes. And also the collaborative investigations, that is very much important. Since the drug development, particularly for an ideal therapeutic agent, is long, expensive, and drudgery process that require multi-disciplinary collaborations among the educational institutions in research laboratories and healthcare professionals. So that's why our Prime Minister Narendra Modi announced the deep sea missions and how we can utilize these things deep sea resources for the betterment of, for the development of various materials in terms of medicines, products, all these things for the well-being of society. So in this case, once the researchers is ready to share our hands from national into the international level instance, definitely we can achieve best. And also the duration, that is very much important. Most of the fellows are regularly asking why the research is not producing a drug for this pandemic coronavirus. My dear friends, this kind of things not happen at a single night. We have to work hard. So that's why all our researchers, they are involving maximum level to developing a drug or molecule for curing this pandemic disease called as the coronavirus COVID-19. Because it's a very long process. It's a very long process, a lengthy process. Almost 10 to 15 years we should involving for this scenario. Then only we can developing a very promising drugs into the society. And these are the informations regarding the general remarks. And what are the challenges and targets? Yes, if you are involving into their research, a lot of challenges and the targets, we have to fix the target, but we are also facing a lot of challenges, starting from the sample collection, screening, revalidation, identification, biosafety, recycling the waste, and cost. So these are the things we should considering while processing or developing the biomolecules or drug from the sea. And also, but we should concentrate the animals or the organisms. What are they? Starting from marine microorganisms, other symbiotic group, or even we are also concentrating the biofuel production in pharmaceutical product, nutraceutical agents or material, and we are also developing some of the nanomaterial. And last but not least, we have developing some of the beneficial material or biomedical material from the waste at the concept of wealth from the waste. So in this aspect, we need a lot of collaborations and coordinations. Almost, we already share our hands into the international platform. Of course, the national funding agencies, that means our government of India, they have provided a lot of support to us. And definitely, with the support of government of India and the collaborations with the international research institute or the universities, we have developing very promising leads or the drugs from the sea or the ocean very future. And all these the credentials or all this output from my laboratory. Recently, Department of Science and Technology from my laboratory and identify my laboratory as National Facility Program. And this is the first national facility programs which are supported by the Department of Science and Technology to Annamalai University. That means 90 years of Annamalai University history at the worth of 3 crores. And recently we have launched the specific website for that, and what is our services and what is who we are and what are the facilities are available, what are the details of our collaborations, and peoples are involving on the same area. So this kind of information on the particular website. And this is the facilities which are available at my laboratory. Maximum, what are the instrumentation facilities which are available? All are in working in condition, and we are ready to provide our services at very cheaper rate when compared to any other institutions, starting from the HPDLC, also the HPLC, such as the analytical as well as the preparative HPLC, and we have the ELISA reader, and recently our laboratory equipped with the LCMS facility. That means the ultra high performance liquid chromatography, LCMS 8040 facilities. This facility is available at IIT Madras and Trichy, and in between these areas, this kind of, especially the triple quadruple LCMS facility is not available, especially in Tamil Nadu. So we have these such kind of facilities. Those who are the researchers are very much interested to do some are working on the LCMS or the chemical fingerprinting, sequencing of your lower molecular peptide instance. You are always welcome, and we are ready to support you. And this is the another other facilities which are available, and we have the antimicrobial screening facilities for the 12 human pathogen facilities, and we are immediately accept the samples for screening the antimicrobial activities, including the bacterial as well as the fungal. And these are some of the informations which are available or which are comes out very significant contributions to the science. And my research output starting from the Nature during 2004 as a PhD student, and then as well as our voyage is going on. And my dear friends, this is my research team. And my name is, as you know, that I am as a principal investigator, and my mentor, Professor Dr. T. Balasubramanian, he is a project advisor, and personally, he is a mentor and dynamic director, former director of our center. And on this happiest occasions, I should thank maximum all the funding agencies, starting from Biotechnology, Ministry of Health Sciences, University Grant Commissions, as well as the DAD. And even recently, I have awarded the Erasmus Fellowship by the Gent University, and I worked at the KDA Center for Biotechnology, Albano University Center, Stockholm. So, my dear friends, I hope that this lecture or the information somewhat create a limelight to this particular area, how we can developing or how we can involve in this area. But once you are ready to involving, or once you are ready to deep in dive, we need patience. We need patience, and we need a lot of calmness and all these things to achieve our level best. So thank you very much.
Thank you, sir. Thank you, sir. Thank you for the wonderful, colorful presentation with a lot of videos and animations. I hope the participants would have enjoyed it very much. We have a few questions, sir, from the participants.
Yes.
One of which is, they would like to know what is the prospect of seaweeds in the future?
Especially for the seaweed, lot of prospect, not only for future, it is also in present scenario, because we can developing lot of materials in terms of organic manure, from organic manure to the drug. Lot of opportunities or lot of promising are there, but we should dealing the seaweeds, targeting our aim. That means targeting our goals. Whether you prefer the molecules development or you can developing the biomaterial development, like that, then you can succeed your goal. So a lot of promising are there.
Okay. And with respect to this luminescent bacteria, sir,
Yes.
Does it have any purpose?
Yeah.
Does it have any importance?
Important for their host animal or important for us?
No, sir. Important for researchers.
Yes. The bioluminescent animal, for example, you can think, once we are developing a bioluminescent protein, and this is my idea, and you can create for the parts. So, though, especially our fishermen, even it's very particular in Tamil Nadu fishermen, they are regularly deal with lot of crises by the neighboring countries. because in sea there is no boundary.
Okay.
So we can creating a natural boundary by the natural material. That is possible. This is the one way for utilizing such kind of the bioluminescent material or bioluminescent protein from the marine microbes.
And another participant would like to know whether there is any medicine that is available for viral diseases from marine products, sir.
Yes. Not why the person asking a question for simple antiviral activities, but we are not dealing with the COVID-19. This is very, very rare, and also most of our laboratory, they are not are the viral working setup. So if you are definitely we are even one of my senior, he did his attempt for developing small molecules from the nudibranch animal for the anti-HIV activities. Now he is working in US. So lot of opportunities are the lot of the molecules or source are available from this.
So we have a participant from West Bengal, Debadatta Das, who is very much interested in your research and would like to join with you in collaboration and do some research. He is asking whether there is a scope for joining together, a collaboration with you.
Yes. All the national as well as the international collaborations, we are very much welcoming you. As per your interest, you are always welcome, and just you can searching the Google search engine, just Arumugam in Marine Innocence, you can easily contact me, and immediately I will replying for your request.
Yes, sir. And some of the students who are doing their master's or degree in pharmacology, they would like to know how useful will the marine biology studies be for pharmacology, sir?
Yeah. And my dear friend, actually, this kind of the developing a lead molecules or the drugs. This is not a one-man show. All the responsibilities. As a marine biologist, we should know how the animals are surviving on the environment. And some of the fellows, for example, the chemistry peoples, pharmacological peoples, biotechnological peoples, even engineering people, they should share the hands. The backbone is the marine biologists, but remaining all the disciplines or the discipline peoples, they are once they are ready to share the hands in the sense, definitely we can give the better products to our society.
Yes. And there is another question, sir. They would like to know the scope of marine biology in the future, sir.
Yeah, my dear friend, and the Center of Advanced Study in Marine Biology, this is the only center as Center of Advanced Study in Marine Biology. We are offering the courses MSc Marine Biology and Oceanography, MSc Marine Biotechnology, MSc Coastal Aquaculture, and MSc Ocean Science and Technology. Especially the MSc Marine Biology and Oceanography, once you have completed the particular degree, our people not only available on the land, our people also available on the space. One of my senior, or one of our senior, he is Professor Kasturi Venkatesan. Now he is working on the NASA space research. He is basically marine biologist. You can imagine how the marine biologist working under NASA. What is the relevance from ocean to space? So all these things, it depends on the interest and this kind of informations. And almost all the marine biologists or the aquaculture or even the biotechnology students, maximum all of our students available on the international platform. They are doing they are doing the research, and they are dealing with a lot of research, especially they are working as a postdoc fellow, researcher, etc. So a lot of scopes and opportunities are available. All these things, it depends on your interest. That means students' interest.
There is another question, sir, regarding the the bacteria from the marine origin.
Yeah.
Are these bacteria culturable in the laboratory for testing and getting novel enzymes?
Yeah, very, very good question. Very good question. And the culturing the bacteria, it is not much possible while in captive or laboratory in condition, especially the marine bacteria. But some other bacteria, they have easily cultured, or we can challengingly culture on their laboratory or in captive condition. That is possible. But we should work on that, how we can culturing the marine organisms in the in captive condition. That is another challenging task for us.
Okay, sir. And there is another participant who has asked about the scope of coral conservation through marine biology, sir.
Yeah. Then once you are what is the researchers or the marine biologists, they should know the biology of coral reefs. And once you should know the biology of coral reefs, definitely you can maintaining their natural diversity of coral reef into the natural environment. That is possible.
So a lot of participants have appreciated the presentation, sir, and they would even like to have the PPT of yours. Maybe they can contact you in your website and request for the same.
Yeah.
Once again, I would like to thank you very much for this wonderful, informative session, sir. I request Maslamani Selvam, sir, to say a few words.
Yeah.
Dr. Maslamani, sir. Maybe his connection is lost.
Yes, sir.
Yes, sir.
Dr. Arumugam, sir.
Yeah.
Hello.
Yeah.
Yes, sir. Yes, sir.
So, very good afternoon to all of you. Dr. Arumugam, sir, you have given an elaborate lecture on drugs from the sea. Thank you very much, sir. So, it's really useful to the participants, all the participants, and very, very thank you, sir. Even though the internet connection is unstable, still you have managed. So, thank you very much, sir. And tomorrow's speaker, Professor Dr. K. Kadaresan, DSc, and he'll be talking on mangroves and aquaculture. Please be on time at 4 o'clock, and it will be very interesting as well as informative. And so once again, thank all the participants for attending this webinar, and see you tomorrow. Thank you.
Thank you. Thank you, sir. Thank you very much once again, sir.
Yeah.
Thank you to all the participants. Please do join us tomorrow. Thank you, one and all.